Assalamu alaikum, dear students. Welcome to the course Principle in Animal Life 1. This is lecture number 59. The topic is various modes of transport across the cell membrane. In this lecture, we will discuss passive transport. Discussing agenda for the topic is passive transport is diffusion of a substance across a membrane with no energy investment. Effects of osmosis on water balance of cells without cell walls, facilitated diffusion, passive transport aided by proteins. Lecture outcome. After watching and listening this lecture, students will know about the passive transport of materials across the plasma membrane. Introduction. Passive transport is diffusion of a substance across a membrane with no energy investment. So passive transport is that type of movement across a plasma membrane where no ATPs are consumed. The molecules are in bulk and they have a tendency to move towards area where their concentration is low. So this movement along concentration gradient, this presence of molecules in a bulk provides the energy for this type of transport. Molecules have a type of energy called thermal energy due to their constant motion, which is also known as Brownian motion. One result of this motion is diffusion, the movement of particles of any substance so that they spread out into the available space. So the molecules tends to move from an area of higher concentration towards an area of lower concentration till a time reaches when the molecules are uniformly distributed in the available space. Then thereafter the movement of molecules also takes place, but then a dynamic equilibrium is established. Each molecule moves randomly, yet diffusion of a population of molecules may be directional in a sense that they move from an area of higher concentration towards an area of lower concentration. Imagine a synthetic membrane separating pure water from a solution of a dye in water. Now study part A and B of figure 1 carefully to see how diffusion would result in equal concentration of dye molecules in both solutions. Much of the traffic across plasma membrane occurs by diffusion because the molecules are in abundance in the extracellular space and this bulk presence of molecules on the outer side of the cell possesses potential energy which draws these molecules inside into the cell. So now here we will discuss these two diagrams. The first one is the diffusion of solute across a synthetic membrane. So this is a synthetic membrane, you can say a semi-permeable membrane, and the molecules of dye are in large concentration on one side of this membrane. So these molecules will have this natural tendency to move towards an area where their concentration is lower, so they will move in the right direction, and here some of the molecules have sneaked into the right side. Now these molecules on the right side, they also show further motion, and some of them will spread in the space, whereas the others will tend to return back via the plasma membrane until a time will reach when the concentration of molecules on both sides will be same, and still there will be movement across the plasma membrane, but there will be no net movement and a dynamic equilibrium will be established. So similar is the case with two dyes. If you take two dyes, then these molecules tend to move towards areas where their concentration is lower, and a time will reach when dynamic equilibrium will be established across both sides of the plasma membrane, and during this process the concentration of the molecules on both sides will be equal. So passive transport is diffusion of molecules from an area where their concentration is higher towards an area where their concentration is lower. So once the dynamic equilibrium is established, then there will be no net movement of molecules. Same number of molecules moving from one side to another, and the same number will also move from that opposite side. So there is a simple rule of diffusion. In the absence of any other forces, a substance will diffuse from where it is more concentrated to an area where it is less concentrated. You can put that other way, that a substance diffuses down its concentration gradient, that is the region along which the density of a chemical substance increases or decreases. So diffusion is a spontaneous process needing no input of energy, and each substance diffuses down its concentration gradient, that is from higher to lower level. So much of the traffic across cell membrane occurs by diffusion because it is a convenient process for the cell and for the organism overall to draw molecules inside the cell where no energy will be required. When a substance is more concentrated on one side of a membrane than on the other side, there is a tendency for it to diffuse across down its concentration gradient. One important example is the uptake of oxygen by a cell performing cellular respiration. So oxygen can directly diffuse into the lipid bilayer, but the only condition is that its concentration must be higher on the outside as compared to inside. Then it will travel inside the cell courtesy of its concentration gradient. Dissolved oxygen diffuses into the cell across the plasma membrane. As long as cellular respiration consumes the oxygen as it enters, diffusion into the cell will continue because the concentration gradient favors movement in that direction. So if a cell is continuously carrying out the process of aerobic cellular respiration, then it means that cell is actively consuming the oxygen which is coming inside, and therefore the concentration gradient will be established because there will always be lesser number of oxygen atoms inside as compared to outside. So the flow of oxygen towards inside of the cell will continue, but a prerequisite condition is that there must be aerobic cellular respiration going on inside the cell. Now, the diffusion of a substance across a biological membrane is called passive transport because it requires no energy. Now, the concentration gradient itself possesses potential energy, and courtesy of that, molecule moves towards areas of lower concentration. Remember though that membranes are selectively permeable and therefore have different effects on the rates of diffusion of various molecules. So those molecules which are in bulk outside of the cell, then another factor which is taken into consideration is the permeability of the plasma membrane to that specific molecule. If the plasma membrane is permeable, then the rate of diffusion will be higher. If the plasma membrane is not permeable or less permeable, then the rate of diffusion of that molecule will be slower. So water can diffuse very rapidly across membrane of cells with aquaporins. These are the channels through which the water molecules enter into the cells. About 3 billion water molecules enters per second via aquaporins, compared with diffusion in the absence of aquaporins. So if the water molecules tends to invade the plasma membrane directly, then of course the rate of movement will be much slower. The movement of water across the plasma membrane has important consequences for cells. Now we will discuss osmosis. So, effect of osmosis on water balance. So what is osmosis? Osmosis is the movement of water molecules from an area of lower solute concentration towards an area of higher solute concentration. Remember, when you are talking about osmosis, then you will have to consider two things. First, the concentration of the solute, and then the water molecules. To see how two solutions with different solute concentrations interact, picture a U-shaped glass tube with a selectively permeable artificial membrane separating two sugar solutions. Now, this is a selectively permeable membrane, and it is separating two sugar solutions. Now, on the right side, the concentration of sugar molecules is higher as compared to the left side. So, the free water molecules are more on this side as compared to this side. So now redefine osmosis. It is the movement of water molecules from an area of lower solute concentration towards an area of higher solute concentration. So what will happen? The water molecules will move towards that direction, and after some time the level of the water molecules will be higher at this end. As a result, the solution with a higher solute concentration has a lower free water concentration. So water diffuses across the membrane from the region of higher free water concentration, that is lower solute concentration, to that of lower free water concentration, that is higher solute concentration. until the solute concentration on both sides of the membranes are more nearly equal. So let's go back to that diagram once again. As you can see here, on this side there are few solute molecules, so more water molecules are free, and on this side there are more solute molecules, so more water molecules are occupied, they are bound with the solute molecules, and less water molecules are free. So now the water molecules on this side, the free water molecules are higher on this side, free water molecules are lower on this side. So the movement of the water molecules will be from an area where their concentration is higher towards an area where their concentration is lower. So redefine this again. So water diffuses across the membrane from the region of higher free water concentration to that of lower free water concentration. So higher free water concentration will be that area which has lower solute concentration, and lower free water concentration area will be that which has higher solute concentration. So this movement will continue until the solute concentration of both sides is equal or nearly equal. So the diffusion of free water across a selectively permeable membrane, whether artificial or cellular, is called osmosis. Now the movement of water across cell membranes and the balance of water between the cell and its environment are crucial to organisms, because the organism must maintain a proper level of different ions in water inside the cell, and for this process the organisms are actively engaged in a mechanism called Osmo Regulation. So let's now apply what we have learned about osmosis in this system to living cells. So water balance of cells without cell walls, that is animal cells. To explain the behavior of a cell in a solution, we must consider both solute concentration and membrane permeability. Both factors are taken into account in the concept of tonicity. What is tonicity? The ability of a surrounding solution to cause a cell to gain or lose water. The tonicity of a solution depends in part on its concentration of solute that cannot cross the membrane relative to the inside of the inside the cell. So in the coming slide we will further clarify this concept. So a solution is isotonic, or a cell is said to be in an isotonic solution when there is no net movement of molecules inside and outside. So water will diffuse across the membrane at the same rate in case of dynamic equilibrium. Now in an isotonic solution there will be no difference in the volume of the cell, because the movement of water molecules towards inside will be equal to the movement of water molecules exiting the cell. So let's consider another situation where a cell is kept in a hypertonic solution, means the solute concentration outside the cell is higher as compared to inside the cell. Now the solute concentration inside the cell is low, so it means free water molecules are in higher number inside the cell as compared to outside. So what will happen? The water will leave the cell and the cell will shrink, and this often occurs in the saline environments, where the salinity of the water is extremely higher and the organism cannot afford to have such salinity inside the cell. Therefore, the organisms living in marine environments have uh, evolved different mechanisms through which they can get rid of excess of salts coming to their bodies with water. And the marine invertebrates are in fact osmoconformers. They doesn't care about maintaining a proper level of water and ions inside the cell. They are isotonic with the surrounding environment. Now, in other cases, in other circumstances, a cell is placed in a solution where the solute concentration of the solution is lower as compared to that inside the cell. So the free water molecules inside the cell are in lesser concentration as compared to outside. So what will happen? The water will start entering into the cell, and as a result the cell will swell and burst like an overfilled water balloon. So here, hypotonic solution, a cell is placed in a solution where the solute concentration outside is lower as compared to inside the cell. So more free water molecules are available outside as compared to inside. So what will happen? The water molecules will rush into the cell and the cell will burst. In the second case, the solute concentration both outside and inside is same. There will be movement of water molecules, but there a dynamic equilibrium will be established, and there will be no effect on the volume of the cell. In the third case, which occurs in the marine environments, where the solute concentration outside is higher as compared to inside, so more free water molecules are available inside, and therefore these water molecules will tend to move outward to the area where the concentration of free water molecules is lower. So the cell will lose water and it will shrink or shrivel. So a cell without rigid cell walls cannot tolerate either excessive uptake or excessive loss of water. So both hypertonic and hypotonic environments pose problems to the cells lacking cell walls. So this problem of water is automatically solved if such a cell lives in isotonic environment. Then of course there will be no need of maintaining a proper balance of water and ions if the outside environment is isotonic to the inside. So seawater is isotonic to many marine invertebrates. So the marine invertebrates are osmoconformers. The cells of most terrestrial, that is organisms that live on land, are bathed in an extracellular fluid that is isotonic to the cells. So in hypertonic or hypotonic environments, that is, hypertonic is marine environment, hypotonic is freshwater environment. So the organisms that lack rigid cell walls, that is the animal cells, they must have certain adaptations which enables them to maintain a proper balance of salt and ions and water inside the cell. And this mechanism to maintain a proper level is called Osmo Regulation. For example, the unicellular protist Paramecium caudatum lives in pond water, which is hypotonic to the cell. So the Paramecium cell has some kind of adaptations to cope with that environment, and the Paramecium cell does not burst because it has a contractile vacuole, and this contractile vacuole pumps excess water outside the cell as fast as it enters by osmosis. In contrast, the bacteria and archaea that live in hypersaline, excessively salty environments have cellular mechanisms that balance the internal and external solute concentrations to ensure that water does not move out of the cell. So now facilitated diffusion. Facilitated diffusion is also passive transport, but it is aided by proteins. Means this transport also takes place without the consumption of ATPs, but instead of passing directly through the plasma membrane, this transport is facilitated by the embedded proteins. Now, some molecules are hydrophilic, and these hydrophilic molecules cannot pass through the plasma membrane directly because the inner of the plasma membrane is hydrophobic. So polar molecules or hydrophilic molecules and ions are blocked by the lipid bilayer of membrane, diffuse passively with the help of transport proteins that span the membrane. So transport proteins are located in the plasma membrane, although the proteins of the plasma membrane themselves are amphipathic. The ends protruding outward are hydrophilic, whereas the main body which resides in the bilayer portion is hydrophobic. But these proteins have internal channels or internal paths which are hydrophilic, and therefore they facilitate the movement of hydrophilic molecules. So this type of diffusion is called facilitated diffusion because this diffusion is facilitated by the membrane proteins. It is aided by the membrane proteins. The cell biologists are still trying to learn exactly how various membrane proteins facilitate diffusion. Most transport proteins are very specific, that is, they are specific for one type of substance. As mentioned earlier in the previous lectures, the transport proteins are of two types, that is channel proteins and carrier proteins. The channel proteins have a hydrophilic channel, and this hydrophilic channel makes a situation comfortable for the hydrophilic molecules to invade into the cells, whereas the carrier proteins, they change their conformation during the process of transportation. The hydrophilic passageways provided by these proteins can allow water molecules or small ions to diffuse very quickly from one side of the membrane to the other side. Aquaporins, for example, are the water channels. They facilitate the massive levels of diffusion of water molecules that occurs in plant cells and in animal cells as well as RBCs. Certain kidney cells also have a high number of aquaporins, allowing them to reclaim or reabsorb water from urine before it is excreted. So if the kidneys do not perform this process of reabsorption, then a person will produce 180 liters of urine per day, and imagine the person will have to take that much of water also to maintain a proper level inside the body. That would have been a cumbersome task. The channel proteins that transport ions are called ion channels. Many ion channels function as gated channels because they are not always open. They are like gates. When someone is waiting outside, then they are opened. When there is no invading molecule, then they are in closed state. But these opening and closing does not occur on its own. There must be some kind of a stimulus. So then there are ion gated channels where the ions provide the stimulus for opening and closing, and then we have the voltage gated channels where the change in voltage provides a stimulus to the opening and closure. So in nerve cells, for example, potassium ion channel proteins opens in response to an electrical stimulus, allowing a stream of potassium ions to leave the cell. This restores the cell's ability to fire again. Other gated channels have a chemical stimulus, so they open or close when a specific substance binds to the channel. Now remember that the substance which acts as a stimulus to the ion gated channels is not the one to be transported. So ion channels are important in the functioning of the nervous system. Carrier proteins such as glucose transporter or GLUT transporter seem to undergo a subtle change in shape that somehow translocates the solute binding sites across the membrane. So such a change in shape may be triggered by the binding and release of the transported molecule. So here in case of the carrier proteins, the molecules to be transported itself act as a stimulus for the change in conformation. So that is contrary to the ion gated channels. Now, like ion channels, carrier proteins involved in facilitated diffusion results in the net movement of a substance down its concentration gradient. Now remember, we are discussing facilitated diffusion, so there is no input of energy required. The energy for the movement of the molecules is provided by the bulk. The bulk possesses potential energy, and as a result of this potential energy, the molecules disperse in space and tends to move towards areas along their concentration gradient where their concentration is lower. So no energy input is required, and thus it is a passive transporter. That will be the end of this lecture. Thank you very much.